Assalamualaikum warahmatullahi wabarakatuh. Hello, good morning ladies and gentlemen. My name is Kholifatul Asysyifa. I am the moderator of Group 2. So, in this video, we're going to give you a short presentation about the controversy of cloning, its pros and cons. But before we start, let me introduce our team. I am Kholifatul Asysyifa as the moderator and the first speaker, Shafira Aulia as the second speaker, Ahmad Syukri as the third speaker, Triantri as the fourth speaker, Amalia Okta Shafira as the fifth speaker, and the last we have Ananda Noviana as the sixth speaker. Our presentation is divided into four sections. First, we will talk about what is cloning and the history of cloning. Second is methodology, so we will discuss about how cloning works. The third is the pros and cons, so we will talk about what is the pro and the cons of cloning. And the last is we will summarize all of that in the conclusions section. Okay, then let's start our presentation. So, what is cloning? According to the National Geography, cloning is a technique that scientists use to create exact genetic replicas of genes, cells, or animals. So basically, creating an exact genetic replicas of living things or organisms. Genes, cells, tissue, or even whole animals can all be cloned, and every single bit of their DNA is identical. Some clones already exist in nature. Identical twins are just one of many examples. They share almost exact same genes because identical twins are created when a fertilized egg splits in two. Next is the history of cloning. The history of cloning is started in the 1885 by a German biologist, Hans Adolf Eduard Dries. So this is the first ever demonstration of artificial embryo twinning. Dries showed that by merely shaking two cells of sea urchin embryo, it was possible to separate the cell, and each cell can grow into a complete sea urchin. Next, in the 1902, a German embryologist Hans Spemann was doing the same experiment but with more complex and stickier embryonic cell from salamander. He is using a strain of baby hair to tighten the cell, and once the cell separated, it can grow into an adult salamander. But when he using the same method to a more advanced salamander embryo, these weren't as successful developing into an adult salamander. So this experiment showed that more complex animal can also be twinned, but only up to a certain stage of development. And in the 1928, again, Hans Spemann, using a strain of baby hair, tried to separate the fertilized egg of salamander. But he temporarily squished the fertilized egg to push the nucleus into the site of cytoplasm. Once it's separated into cell, each cell can be new embryo of salamander. This is inspire other scientists to do experiment and study of cloning using the same method with a different kind of animal like frog, mammalian, rabbit, sheep, cow. So in the 1996, we have the first mammal created by somatic cell nuclear transfer named Dolly. After 277 attempts, her arrival made a controversy of human cloning. So in the 1997, there are scientists that make the same experiment but with primates, rhesus monkey. And this experiment showed that primates, the closest humans relative, can also be cloned. Until now, scientists still doing experiment for a better study and research of cloning, but we still didn't have any proof that there is scientists ever created a human clone. Well, let's move on to the next section. Our next speaker, Shafira Aulia, is going to talk about methodology.
Okay, next methodology. Methodology in cloning, there are two techniques. One, cloning plant and 2 cloning animal. In cloning plant, genetic modification can be used to produce plants that improve food production. There are 2 techniques of cloning plant: cutting and tissue culture. In cutting, the simplest way to clone a plant, a branch from parent plant is cut off. Its lower leaves are removed and stem is planted in damp compost. The cutting is usually covered in a clay plastic bag to keep it moist and warm. After a few weeks, new roots develop and a new plant is produced. Then tissue culture. Tissue culture, this method is more difficult to do than taking cutting. Tissue culture, also called micropropagation, it works with small piece of plant, called eggplant, or a small piece taken from a plant. These are grown in vitro, or experiments are done in glassware such as test tube and petri dish, using sterile agar jelly that contain plant hormone and nutrients.
Cloning animals. The cloning technique in animals, called artificial embryo cloning and somatic cell nuclear transfer, is a cloning technique that developed from reproductive cloning. There are 2 techniques of cloning animals. The first is artificial embryo cloning. This technique mimics the natural process that creates an exact genetic copy or clone of an individual. The second is somatic cell nuclear transfer. This technique, also called nuclear transfer, to make a clone, scientists transfer the DNA from an animal somatic cell into an egg cell that has had its nucleus and DNA removed. There are 3 types of artificial cloning. The first is gene cloning. Gene cloning is also known as DNA cloning, produce copies of genes or segments of DNA. At its most basic, cloning genes in the lab is easy. Combining the proteins that cells use to copy their DNA and adding a gene to copy their genetic material. The cell grows and divides. They naturally create clones using cellular division, a process called mitosis. The second is reproductive cloning, produce copies of whole animals using the somatic cell nuclear transfer technique. This technique is used by Wilmot and Kate Campbell to make dolly sheep. The third is therapeutic cloning, produce embryonic stem cells for experiments aimed at creating tissues to replace injured or diseased tissues. Somatic cell nuclear transfer. Dolly sheep. She was born in 1996 after scientists figured out how to remove the DNA from the egg cell of a Scottish blackfish sheep and basically replace it with the DNA of a mammary cell from a fin dorsal sheep. They gave it a little electric shock to fuse the cell and get it replicating. Place the cell in the uterus of another sheep and boom, a clone.
Next, therapeutic cloning is almost identical to reproductive, except the cloned embryo is never implanted in a uterus. Instead, the embryo is cloned for the sole purpose, extracting stem cell. Next, number 3, pro and conclusion cloning. The first study and research. For study and research on cloning, that is, scientists also make clones in the lab. They often clone genes in order to study and better understand them. The second medication for treatment cloning, that is, cloning can create layers of amounts of protein for medication like insulin or used to be detect the presence of specific strand of DNA, like in the COVID-19 PCR test. Next, regarding increase and production cloning. Livestock like cows and pig have been to produce more milk or meat. Cloning might one day bring back species like in the wool, hammock, again panda.
The cons. We have been able to clone human embryos for about 8 years. But as far as we know, no one actually cloned a whole person. Turns out, ethics aren't the only thing holding scientists back. Number 1. The process is not entirely safe. Most of clones have had reduced longevity and develop health problems. Number 2. The probability of abuse is very high. In the case of Dolly the sheep, it took 277 tries for scientists to get one Dolly. In her, the scientists made many varied attempts. Number 3, the offspring lack genetic uniqueness. And number 4, it is not yet fully developed reference. Until recently, many scientists would agree that the cloning process has not yet been fully developed for use.
Conclusion. Cloning describes the process used to create an exact genetic replica of another cell, tissues, or organism. The copy material, which has the same genetic makeup as the original, is referred to as a clone. The most famous clone was a Scottish sheep named Dolly. There are 3 different types of cloning. Number 1 is gene cloning, which creates copies of gene or segment of DNA. Number 2, reproductive cloning, which creates copies of whole animal. And number 3, therapeutic cloning, which creates embryonic stem cell. Researchers have to use this cell to grow healthy tissues to replace injured or diseased tissue in human body. Cloning needs a lot of consideration, research, and development. Cloning isn't the sci-fi marvel we think it is. It can be dangerous, often ineffective, and most of all, we just haven't thought of a good enough reason to do it. So, cloning might actually have a bigger place in a movie than does in real life, because the money just isn't there. And just because we can do something doesn't mean we need to. And that brings us to the end of the presentation. Thank you so much for your interest and attention. Wassalamualaikum warahmatullahi wabarakatuh.